Hi guys, today we are talking about um, gene regulation, specifically in prokaryotes. Um, but first, I wanted to talk a little bit about the differences between prokaryotes and eukaryotes. And this is definitely a continuation of the last video, which was related to transcription and translation. Um, this video is going to be related to how prokaryotes control which genes get transcribed and translated at specific times. So first of all, as a little bit of review and a little bit of newness, prokaryotes have a much smaller genome, which means they have fewer genes. As a reminder, the genome is all of the DNA of that organism. So for bacteria, which is what prokaryotes are, they have fewer genes and a smaller genome. So that means that they have higher gene density, which means that they have more genes located in a smaller section of DNA. So for us and for other eukaryotes, we have a lot of sections of our DNA that, number one, we don't know what they do. And number two, they might not do anything. So those are the introns that I was talking about from the last video that are non-coding, which means that they're actually like cut out and not used to make the protein at all. So prokaryotes, because they have higher gene density, are going to have a lot fewer of those non-coding regions, if any. All right, so with that, we're going to talk today about gene regulation for these prokaryotes. And they use something called operons, which this is going to be probably a new topic for you. It's not something that you learned about in freshman bio. So try to be patient with yourself as you learn about this because it is new and it is complex. So to start off, bacteria are often living in these like erratic and rapidly changing environments, which means that they need to have easy ways to really quickly turn off or turn on specific genes. For us, things are less erratic, so we don't usually need to be able to like activate or inactivate huge numbers of genes at a certain time. We do have ways to regulate gene expression, but typically not this fast. So what an operon is it's a DNA section that's regulated by a repressor, which can turn off the promoter site, which is where transcription starts, and that prevents transcription from happening. So both of the examples that I'm going to talk about today are using repressors, which a repressor's job is to turn something off. I'm going to make a note of that here, that repressors turn off transcription. So they make it so that the genes are not expressed. If the genes aren't if the genes aren't expressed, what that means is that they're not being transcribed and therefore they're not being transmitted. So an expressed gene means a gene that is being used to make a protein. Okay, operons contain several related genes, which each have their own start and stop codon. So they're all going to be responsible for like the breakdown or take up of a specific thing. They're all kind of working together and they're all necessary to make the organism like switch gears. And then what's nice is that the operon has only like one on and off switch. So even though there's many different genes in the operon, there's only one like switch to turn them all on or to turn them all off. So it's either all of them are functioning and being expressed or none of them. Okay. So there's two types of operons. They're called repressible and inducible. Both types of operons contain a promoter region, which means that it's the attachment point for RNA polymerase and an operator for the repressor to attach when not needed, and then the genes also for a specific job. So the promoter is like the very beginning of the operon. Repressors are coming from a regulatory point that's away from the operon. They're proteins. And then the two types of operons that we're going to talk about today are repressible and inducible. So repressible means that they are usually turned on, but they can be repressed or turned off. So I'm going to just make a note of that here, mostly because it helps me. So repressible operons are usually on, but can be turned off. On the flip side, inducible operons are usually off, but can be turned on. And both of these are going to be related to repressors to start. And then at the end, I'll talk about one inducer. But first, we're going to talk about repressor proteins as a way to turn both on and off specific operons. There's examples of both of these that we're going to talk about today. An example of a repressible operon is the trip operon. So usually the repressor is inactive until it binds to a tryptophan molecule. And when it binds to a tryptophan molecule, the repressor will become active and the operon will be turned off. Inducible operons, the example we're going to talk about is LAC, the LAC operon. Typically, the repressor is active and bound to the operon, but when the repressor binds to allolactose, it becomes inactive and that allows the operon to turn on. I'm going to talk about both of these using images. Okay, so here's the trip operon. It's related to its ability to, for the bacteria's ability to use and manage tryptophan, which is an amino acid. So what you have is up at the top, the whole operon, and it is all DNA. It's just blocked off to show you the different genes. So at the very beginning, you have the promoter, which is the start of the operon. And you have a regulatory gene that is responsible for producing the actual repressor protein. So that's what's happening on the very left, is that separate from the operon is the code that's needed to produce the actual repressor gene, repressor protein. Then further downstream, which is what we call that, there is another promoter that includes an operator, which is the location where the repressor eventually will bind, and RNA polymerase kind of ready to take off and do transcription. Inside the operon, there are many different genes that are responsible for the production of tryptophan. So normally, the bacteria is producing tryptophan. It's using the enzymes to make that happen. It is doing transcription because nothing is blocking the operator. So it's RNA polymerase is able to just straight on go for it. The red subunit over to the side is the repressor. You can see that it's inactive because the shape of the top here, so that does not fit the shape of the operator. And it's just like enzymes, how enzymes have to be the right shape at the active site to fit with the substrate. Same thing here. So since this repressor is not the right shape to fit in the operator, the operon is active and it's turned on. So now what can happen is this operon can become repressed or turned off. And you would want to turn it off because the cell has enough tryptophan present that it doesn't need to be producing more. So what happens is some of the tryptophan that is present in the cell binds to the repressor, which is happening down here. And that causes the repressor to change shapes and become active. Because it changes shapes, it can now fit into the operator on the operon, which means that RNA polymerase can't zoom through that and continue to do transcription. It gets stuck and no transcription occurs. Eventually, the tryptophan will release from the repressor will become inactive again, and the cell will start to produce more tryptophan. And that will happen once the tryptophan levels get low in the cell and the cell needs to produce more. So in summary, this is a repressible operon. Repressible. And it's only off when tryptophan is present. And the rest of the time, it is on and active. Now let's talk about one that's inducible. So down here is the lac operon. The lac operon is usually turned off because cells prefer to use glucose to do cell respiration and produce energy. But in the case where there is not enough glucose present, the cell actually can use lactose for that. So what happens is you have typically your active repressor, which again was coded for by a regulatory gene separate from the operon. So you have your repressor that fits into the operator. It's active. It's preventing RNA polymerase from transcribing the genes. Now, there can be times where the inducer, which in this case is allolactose, is present. It binds to the repressor. It causes the repressor, in this case, to become inactive. So the repressor falls off or doesn't attach to the operon. And in that case, RNA polymerase can do its job. It can produce these different enzymes that are responsible for helping to break down and digest lactose in the cell. So this is an inducible operon. And it's only on when cell needs to digest lactose. And how it gets turned on is the repressor becomes inactive. Whereas up here, the repressor becomes active. So both of these examples were related to repressors being inactive or active. There's one more example that I'm going to talk about, and that is positive control, which means instead of using a repressor, we're going to use an activator. So bacteria, like I said before, prefers to use glucose to do cellular respiration and produce energy. Just because lactose is present doesn't necessarily mean that the cell wants to use it. It only wants to use it when glucose is low. So there is a positive gene regulator that can also make it more likely that the lac operon will be transcribed. And that actually uses cyclic AMP or CAMP, which we've talked about a little bit before during the cell cycle. So what can happen is cyclic AMP binds to a regulatory protein called CAP, catabolite activator protein. And that complex becomes an activator that binds just in front of the promoter upstream. And this activator makes it more likely that RNA polymerase will attach to the operon and start. So when you have positive gene regulation, you have this cyclic AMP CAP complex, and you also have a inactive repressor that says, okay, lactose is present in the cell, it can be used. And then the CAP is saying glucose is not here, we need to use the lactose. So it's just increasing the chances that the operon will be transcribed. Here's an image of that. So what you have here is still the lac operon. You have your cyclic AMP, which is that golden triangle, binding to an inactive CAP protein to make it active. You can see the shape change slightly when CAP became active. It binds just at the very beginning of the promoter, and that triggers the RNA polymerase to be more likely to start transcription. At the same time, the lac repressor is also inactivated. So it's down here. It says lactose is here, so we're inactive. This is saying lactose present. So transcribe. This is saying glucose absent. So transcribe. So both of these things together are saying like, yes, 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 go, go, go. We want to use the lactose. Now, sometimes what happens is lactose is present, but there's still enough glucose in the cell that the CAMP levels are low. So that's happening down below where you have inactive lac repressor. So this is saying lactose present. So transcribe. But because there's no cyclic AMP, CAP is still inactive. So this is saying we have glucose. Not urgent. So without CAP, RNA polymerase still can bind, but it's less likely to bind because CAP isn't there at the promoter. And CAP is kind of like the push for RNA polymerase to bind to the promoter and start transcription. So some lac mRNA will be synthesized because the block of the repressor isn't there. The operon has been induced. But at the same time, CAP isn't there to give that little extra push. So this is an example of positive control, the cyclic AMP and CAP situation. So as a summary, lactose and tryptophan are both examples of operons in prokaryotic cells. Lac is an inducible operon, which means that usually it's off, except when lactose is present, and then the repressor becomes inactive and the gene can be transcribed. Tryp is usually on, so usually the cell is producing tryptophan. When there's enough tryptophan present, the operon becomes repressed because tryptophan binds to the repressor, changes its shape so that it binds to the operator of the operon. That blocks transcription so that it turns off. There also are situations where activators can be used to increase the likelihood of the operon being transcribed. And one example of that is CAP and cyclic AMP when glucose levels fall in the cell. All of this is only for prokaryotic or bacterial cells. Eukaryotes like us do this a little bit differently. We don't use operons because we don't need to turn on a whole bunch of genes all at one time really fast. We'll talk more about eukaryotes next time. Thank you. Good job. See you soon.